Hello friends, dear learners and my dear student. First of all, I take an opportunity to welcome you at my M.Sc Analytical Chemistry class. Friends, I am Dr. S.R. Labhade, working as Associate Professor in Chemistry at KTGM College, Nasik. Student, in previous lecture we discussed the theory and analytical principle of cyclic voltammetry. We also discussed the electrode system employed in cyclic voltammetry. In addition to this, we also discussed the potential sweep program employed to the working electrode in cyclic voltammetry. In a positive scan, we are increasing the potential from negative to positive in order to carry out the oxidation of an analyte. And in negative scan, we increase the potential from less negative or positive to negative in order to carry out the reduction of the analyte. Student, in today's lecture, we will discuss the working of cyclic voltammetry. Student, we know that cyclic voltammetry is an important tool of qualitative and quantitative analysis in which cycling of potential to the working electrode is carried out and resulting current is measured under diffusion mass transfer phenomenon. Similar to polarography, cyclic voltammetry is a linear scan potential sweep voltammetry performed in a quiescent or undisturbed analyte solution containing excess of supporting electrolyte. Polarography is a linear scan voltammetry. It is performed in a quiescent analyte solution. Similarly, cyclic voltammetry is a linear scan voltammetry. It is also performed in a quiescent analyte solution. In polarography, analyte solution, analyte is reduced at DME working electrode and resulting reducing current. It is called as cathodic current. Cathodic current is measured for analyte determination. Only cathodic current is measured in a polarographic analysis. In cyclic voltammetry, analyte is reduced or as well as oxidized at a stationary working electrode and resulting cathodic and anodic current is measured. That means in polarography only reduction current is measured by reduction of the analyte. But in cyclic voltammetry, both reduction current, that is cathodic current, as well as anodic current is measured by reducing and oxidizing the analyte. Polarography is performed by using dropping mercury electrode as a working electrode while cyclic voltammetry is performed by using any stationary electrode. Now we discuss the working of this cyclic voltammetry with example of a reducible ferrous-ferric system. We discuss the working of cyclic voltammetry using an ferrous redox system. That means here Fe3 plus is a ferric ion and Fe2 plus is ferrous ion. We can discuss the working of cyclic voltammetry with reference to conversion of ferric ion to ferrous ion and conversion of ferrous ion to ferric ion, that is a ferric ferrous redox system. Now for this study, the oxygen-free Fe3 plus, that is analyte, is with excess of supporting electrolyte is taken into a voltammetric cell containing stationary working electrode, auxiliary electrode and a reference electrode. That means in polarography, essentially we have to remove the dissolved oxygen in order to avoid the reduction of oxygen at working electrode. Similarly here we have to remove the dissolved oxygen from analyte solution. Therefore oxygen-free Fe3 plus ion as an analyte taken in presence of supporting electrolyte like KCl into voltammetric cell containing working electrode, auxiliary electrode and a silver chloride or saturated carbon electrode as a reference electrode. In previous lecture we discussed the potential sweep program applied in a cyclic voltammetry. Generally in cyclic voltammetry, initially the potential of the working electrode is kept at a zero value in order to avoid any oxidation or reduction reaction. At the beginning of the experiment, the potential of working electrode is maintained at zero value to avoid any reduction, any electrochemical reaction, that is oxidation or reduction, to avoid any electrochemical reaction of analyte. In this case, in this study we take Fe3 plus as an analyte. Fe3 plus is present in its oxidized form. That means this will be get reduced. And in this study, the first scan, that is a forward scan, in forward scan we have to increase the potential from less negative to more negative in order to carry out the reduction of the Fe3 plus. In this study, Fe3 plus is essentially essentially to reduce at working electrode. So the forward scan is the negative scan. That means here we can increase the potential of working electrode from less negative to more negative, so that Fe3 plus is reduced. And during this we measure the cathodic current. Therefore, Fe3 plus electron gives Fe2 plus. Therefore, when Fe2 plus is formed, definitely the second scan or reverse scan, the reverse scan is the positive scan, in which we are increasing the potential from more negative to less negative or positive. We increase the potential of working electrode from more negative to less negative or positive. So that the analyte is get oxidized. Fe2 plus gives Fe3 plus electron. If we consider here Fe3 plus as an analyte, then forward scan is a negative scan in order to reduce Fe3 plus to Fe2 plus. And reverse scan is a positive scan in which we can oxidize Fe2 plus to Fe3 plus. Now in this lecture we discuss the potential sweep program using cyclic voltammetry, that is in a triangular waveform. In this case here we are increasing the potential of working electrode up to certain level, and after that we can reverse that potential again to its original line. Here this is a graph of time in second, here 0, 40, here 20. And when time reaches to 20 or when a first scan is completed, we can reverse that scan. Then this potential is called as switching potential. This is a switching potential at which we can reverse the scan. Therefore, forward scan is here a negative scan. This is a potential, suppose minus 0.2, minus 0.4, minus 0.6, minus 0.8, minus 1.0 volt, like this. Now this is a potential sweep program used in cyclic voltammetry. That means initially we can scan the potential to more negative direction. That may be a first scan or forward scan, and after switching potential we can scan that potential in opposite direction to its original value. Therefore, triangular waveform is a potential sweep program, and that is applied in a cyclic voltammetry. Now we discuss how this potential sweep program gives the cyclic voltammogram. Similar to polarigraphic analysis, the result obtained in cyclic voltammetry is a cyclic voltammogram. It is again a graph of cathodic and anodic current as a function of applied potential. Therefore, here in this case, forward scan, that is a reduction of analyte Fe3 plus to Fe2 plus, and that will be takes place in this case. Therefore, here we can show this forward scan with this yellow color line. Therefore, here as we are increasing the potential of this working electrode, suppose this is a point A, this is point B, this is point C, and this is point D. As we are increasing the potential of working electrode, initially we observe very small current flowing through this cell. That is similar to the residual current in polarography. When this potential reaches equal to the decomposition potential, after that we can observe the current flowing through the circuit. Then here at this point A, initially potential is 0. That means there is no analyte reaction. But when potential reaches to sufficiently high, that is at point D, which is equal to the decomposition potential of analyte, after that analyte starts to undergo reduction at working electrode. And here, as reduction of analyte takes place, here we observe the maximum current. The current decreases to 0 value when total analyte is converted into the product. Therefore, here in this case, Fe3 plus is totally converted to Fe2 plus by reduction process, and that is shown by this point. Here, as the potential of working electrode we are increasing, therefore at a particular point we observe the increase in current. That is because of the reduction of the analyte. Therefore, this is a point B here. This is a switching potential. Switching potential. Similar to this is a potential axis, and here this is a graph of current, which is a cathodic current because it is obtained due to reduction of analyte. This is I in microampere due to the oxidation of analyte. Therefore, cathodic current due to reduction of analyte and anodic current due to oxidation of analyte. Initially, this current is 0 when potential is 0. It will be increased to maximum 80. Here it is 40, here it is minus 40, and here it is minus 80 millivolt. Sorry, not millivolt, microampere. Therefore, in this case, in forward scan, as we are increasing the potential of working electrode linearly to more negative direction, here at point A, potential is 0, no reduction of analyte takes place. When it is equal to potential equal to B, at that time it is a decomposition potential of analyte. This is the small current because of the supporting electrolyte, and that is called as residual current. When analyte reaches to its decomposition potential, after that there is sudden increase in current because of reduction of analyte. As total analyte is consumed or it is converted into the product, current is decreases suddenly to 0 value, and further increase in potential we observe little bit increase in current. That is because of reduction of the supporting electrolyte. As supporting electrolyte, reduction of supporting electrolyte starts, we have to change the potential, or we have to reverse the potential. That is reduction of supporting electrolyte that signals for the switching potential. Therefore, we can switch that potential in opposite direction. That means here in second case we can switch that potential from this value to this value to again original 0 value. Now during this what happens here, again we have here E, here F, here G, I. Now here, when the potential of working electrode is scanned in opposite direction, again we have another wave to the counter side of this cathodic current, and that is because of the oxidation of analyte. Now this is another wave obtained when we scan the potential in opposite direction. This is a point E and this is point F. Here this is point F, this is point G, and this is point R. Such a type of voltammogram is obtained on a graph paper, and that is called as cyclic voltammogram. First wave in voltammogram, that is because of reduction of Fe2 plus to Fe3 plus. And second wave here because of oxidation of Fe2 plus to Fe3 plus. Therefore, in forward scan, that is called as reverse scan, in forward scan, that is called as negative scan, the reduction of analyte takes place. And we are measuring the reduction current as a function of applied voltage. And in second case, on in reverse scan, we oxidize that analyte by reversing that potential, and in this case, we can measure the anodic peak current. Therefore, in voltammetry, cyclic voltammetry, both cathodic current and anodic current is measured as a function of voltage. That is, in forward scan, one current is measured, in a reverse scan, another current is measured. That may be cathodic or that may be anodic. Depending upon the course of reaction, forward scan may be negative, or sometimes it may be positive, or reverse scan may be positive or it may be negative. But when we scan the potential from less negative to more negative, that is termed as a negative scan. And when we scan the potential from more negative to less negative, that is called as positive scan. In a reduction current, we have a positive current, and in an oxidation current, we have a negative value of current. Therefore, here both cathodic as well as anodic current is plotted as a function of applied EMF, and a cyclic voltammogram is obtained. In this case, the peak at which this the potential at which the first peak is obtained, this potential is called as EPC, and the peak at which we have is EPA. Therefore, EPA and EPC, these are the potential at which we observe two currents. One is because of reduction of analyte and another is because of oxidation of analyte. Here EPA and EPC, these are the two parameters obtained on a cyclic voltammogram. Again, if we draw the baseline, and if we determine the peak height of these two peaks, this peak height is called as IPC and this peak height is called as IPA. This is IPA and this is IPC. The result obtained in cyclic voltammetry is in the form of cyclic voltammogram. That is a plot of cathodic and anodic current as a function of applied potential. From the cyclic voltammogram, we have four important parameters, that is IPC, cathodic peak current, IPA, anodic peak current, EPC, cathodic peak potential, and EPA, anodic peak potential. Here we discuss the importance of these four terms and how those are utilized for a qualitative and quantitative analysis in cyclic voltammetry. First of all, first parameter is a cathodic peak current. Cathodic peak current, it is shown by IPC. That is the magnitude of magnitude of current obtained because of a reduction of oxidant, and here oxidant means Fe3 plus ion. Therefore, IPC is directly proportional to concentration of oxidant, and here in this case IP is directly proportional to concentration of Fe3 plus. That means we can determine the concentration of Fe3 plus in the solution by measuring the cathodic peak current. Similarly, second parameter is a anodic peak current, and it is denoted by IPA. IPA is the magnitude of current obtained because of oxidation of reductant, and therefore here this IPA is directly proportional to concentration of a reductant. That means here IPA is directly proportional to concentration of Fe2 plus. A reductant means initially it is present in reduced form and it is oxidized. Here oxidant initially present in reduced form and initially present in oxidized form, and furthermore it is reduced. Reductant initially present in reduced form and it is then oxidized. Therefore here concentration of both oxidant as well as reductant that can be measured from the respective peak current. Another parameter in the cyclic voltammetry is the cathodic peak potential and anodic peak potential. Third parameter is cathodic peak potential, that is EPC. Cathodic peak potential is the potential at which cathodic peak current is observed. Cathodic peak current is observed. And next parameter is anodic peak potential, that is EPA, is the potential at which the anodic peak current is observed. Therefore, in cyclic voltammogram, EPC, this is EPC and this is EPA. EPC and EPA is separated by a factor 50 millivolt when a 1 electron redox reaction taking place. Therefore, here in cyclic voltammetry, the EPC or EPA and EPC are separated by a potential 57 millivolt for a redox reaction with 1 electron. That means here this peak will be observed, that is a 28.1 beyond to a half wave potential, E1 half, and this peak is like before 28.5 millivolt before the half wave potential. Therefore, the central potential is the half wave potential, which is a identity of analyzed species. That means here these two peaks are separated by 57 millivolt. At the center there is half wave potential. Therefore here half wave potential is at the center. Therefore a cathodic peak potential lies beyond 28.5 millivolt and anodic peak will be lies before 28.5 millivolt. Therefore here this analytical information we can obtain from a cyclic voltammetry. That means here IPC is directly proportional to concentration of species undergo reduction. IPA is directly proportional to concentration of species undergo oxidation. Similarly, EPC and EPA, those are separated by 57 millivolt, and at the center there is half wave potential, which is the identity of the material. Student, this is all about the working of cyclic voltammetry and the applications of cyclic voltammogram in qualitative and quantitative analysis. Based on our today's discussion, you can find out the answers of following question. Question number 1. Explain the working of cyclic voltammetry with suitable example. Second, draw the schematic diagram of cyclic voltammogram and explain the following terms. First one is called as cathodic peak current, anodic peak current, cathodic peak potential and anodic peak potential with reference to their analytical importance. In addition to this, you can write out the answers how cyclic voltammetry or cyclic voltammogram is used for qualitative and quantitative analysis. Student, with our today's discussion, you can find out the answers of this question. In forthcoming lecture, we can discuss the different analytical applications of cyclic voltammetry, how cyclic voltammetry is useful for the study of the electrochemical reaction, how we can judge the reversible and irreversible reaction or quasi-reversible reaction by cyclic voltammetry. We also discuss the number of electrons involved in redox reaction. Therefore, with this, we stop here. Thanks for joining me. Wish you all the best. Keep learning. Happy learning.